close your eyes, if you are still reading these words with them open, and let them fall gently closed now, like two soft curtains drawing slowly across a window at the end of a long, long day. Let your breathing slow, just a little, and let the weight of your body settle more fully into whatever surface holds you at this moment. Whether that is a mattress, a pillow, or the warm and familiar contours of a chair that has known you for years. There is nothing you need to do tonight. There is nowhere you need to go. There is only this voice and this story and the quiet dark around you and the long, slow river of history that we are about to drift down together, very gently, with no particular hurry. If this voice helps you rest tonight, consider subscribing to Plague Lullaby, but only if you are still awake enough to remember to do so. And if you are not, then that is perfectly fine, because that means it is already working. Tonight, we are going to talk about a mushroom, a small, pale, quietly beautiful mushroom that grows beneath oak trees, in dappled woodland light, in places that smell of damp earth and autumn rain and the slow, patient turning of seasons. It has a greenish-yellow cap, streaked faintly with olive or white, and it rises from the forest floor on a slender, pale stem, wearing at its base a small white cup that mycologists call a vulva, like a little cradle or a little shroud, depending on how you choose to look at it. It is called, simply and accurately, the death cap, and its scientific name is Amanita phalloides, a name given to it in the early 19th century by the botanist Elias Magnus Fries, who catalogued it with the careful, methodical attention of a man who understood that some beautiful things must be named precisely, so that the naming itself serves as a warning. For thousands of years before Fries gave it its Latin designation, this mushroom had been killing people, quietly, slowly, with a patience that seems almost botanical in its stillness. And tonight we are going to follow that long, unhurried story from its earliest appearances in the ancient world, through the deaths of emperors and popes, through the dark corridors of medieval medicine, through the emergence of modern toxicology, and all the way to the present day, where the death cap continues its quiet work in forests and parks and backyards, spreading slowly across continents it was never meant to inhabit. And we will do all of this slowly, gently, without urgencies, the way a good story ought to be told at the end of a long day. It is worth understanding, before we begin, what kind of thing we are actually speaking of when we speak of a mushroom, because the word itself is one we use so casually, so domestically, so associated with grocery stores and pasta sauces, and the comfortable ordinariness of dinner, that we may have forgotten how strange and ancient and genuinely alien the kingdom of fungi actually is. Fungi are not plants, and they are not animals, and they occupy a kingdom of their own in the classification of life, a kingdom that is in some ways more closely related to animals than to the green and photosynthesizing world of plants, though it resembles neither very much at all. The mushroom that we see above the ground, that pale and delicate structure with its cap and its stem and its soft, papery gills, is only the fruiting body, the visible tip of a vast and largely invisible network of thread-like filaments called mycelium that spreads through the soil beneath it, sometimes for enormous distances, sometimes across acres of forest floor, connecting the roots of trees and exchanging nutrients in relationships that are ancient beyond easy comprehension, relationships that were forming in the deep soil of the Devonian period, more than 400 million years ago, long before the first dinosaur, long before the first flower, in a world that would have been unrecognizable to us in almost every way. The fruiting body, the mushroom itself, appears for only a brief season, releasing its spores and then collapsing and returning to the earth, while the mycelium beneath continues its patient and silent work through the years and decades, waiting for the right conditions to fruit again, waiting with a stillness that has no real analogue in the animal world. The death cap is one such fruiting body, appearing most reliably in the late summer and autumn months in the northern hemisphere, though in places with mild and wet winters, it may fruit at other times as well, and it has been fruiting in European woodlands for an almost immeasurably long period of time, long before any human being walked into those woodlands with a basket, looking for something to eat. The relationship between human beings and mushrooms is itself an ancient one, stretching back to the very earliest periods of human prehistory, to a time when our ancestors were moving through landscapes of forest and grassland and learning, through long and sometimes costly trial and error, which of the things growing around them could sustain life and which could end it. The Romans and the ancient Greeks both recognized the death cap as a deadly poison, and there are references in classical texts to mushrooms that bring death not immediately but slowly, over a period of days, which is entirely consistent with the clinical course of Amanita phalloides, poisoning, as we understand it today. The Greek physician Dioscorides, writing in the 1st century of the Common Era, in his great pharmacological compendium, Materia Medica, described poisonous mushrooms and their effects with some accuracy, noting that certain species brought on a kind of progressive internal collapse that medicine of the time had no means to reverse. It is a testament to the long reach of this mushroom's influence that Dioscorides was writing about it at a time when the Roman Empire was at the height of its power, when legions of soldiers were marching across half the known world, when poets were composing verses that would survive for 2,000 years, and when, in the palace of the emperor himself, the death cap was about to play a role in one of the most consequential and debated deaths in all of Roman history, a death that would reshape the empire and set in motion a chain of events whose echoes historians are still tracing today. We will come to that death in a moment, as we come to all things in this story, slowly, gently, taking our time, letting the details accumulate, the way leaves accumulate on the forest floor in autumn, quietly, one after another, until the ground is soft and deep and ready for sleep. But first, let us understand more fully what the death cap actually is, and why it is so extraordinarily and reliably lethal. Because the mechanism by which this mushroom kills is one of the most elegant and terrible pieces of biological machinery that nature has ever produced. And understanding it, even loosely, even in the soft and unhurried language of a story told at bedtime, will help us appreciate why so many centuries passed before physicians had any meaningful tools with which to address it, and why even today, in an era of liver transplantation and intensive medical care, a meeting with Amanita phalloides remains a deeply serious matter. The death cap contains 3 main groups of toxins, named amatoxins, phallotoxins, and virotoxins, and of these 3 it is the amatoxins, and in particular, a specific compound called alpha-amanitin that are responsible for the overwhelming majority of its lethal effects. Alpha-amanitin is a cyclic peptide, a small, ring-shaped molecule of extraordinary chemical stability, and this stability is one of the qualities that makes it so dangerous because it cannot be destroyed by cooking. It cannot be neutralized by freezing. It survives drying and boiling and any ordinary culinary process that might render a biological toxin harmless, so that a mushroom prepared with care, and even with genuine culinary skill, is no less deadly than one eaten raw from the forest floor, and the unfortunate cook who prepares it believing it to be an edible species, will have no indication until many hours later that anything has gone wrong at all. Alpha-amanitin works by binding to and inhibiting an enzyme called RNA polymerase II, which is the molecular machine responsible for transcribing the DNA of a cell into messenger RNA, the intermediate molecule that carries the genetic code out of the nucleus and into the cytoplasm, where it is translated into proteins. And without functioning RNA polymerase II, cells cannot make new proteins. And without new proteins, cells cannot carry out their normal maintenance and repair functions, and they begin, slowly and systematically, to die. The liver is the organ most severely affected by this process, and the reason for this is both anatomical and biochemical. When alpha-amanitin is absorbed from the gastrointestinal tract, it enters the portal circulation, the network of blood vessels that carries blood from the gut directly to the liver before it passes into the general circulation. And the liver, as the primary organ of metabolic processing and detoxification, receives this first and heaviest dose of the toxin and bears the greatest burden of the damage. Liver cells, known as hepatocytes, are among the most metabolically active cells in the body, constantly synthesizing proteins, processing nutrients, manufacturing bile, breaking down waste products, and carrying out hundreds of biochemical reactions simultaneously. And this high metabolic activity means that they have an unusually high demand for functional RNA polymerase II, which means that they are unusually vulnerable to a toxin that blocks it. The kidneys are the second most affected organ for similar reasons of exposure and metabolic demand, and the clinical course of severe Amanita phalloides poisoning typically involves the progressive failure of both organs, one after the other, over a period of days that has been described in the medical literature as proceeding in 3 distinct phases, each of which has its own particular character and its own particular cruelty. The first phase is a latency period of remarkable silence, lasting between 6 and 15 hours, and sometimes as long as 24 hours after the mushroom has been eaten, during which the person feels entirely well, notices nothing unusual, and may have no reason at all to suspect that anything is wrong. This silence, this deceptive calm, is one of the most medically significant features of the poisoning, because it means that by the time symptoms appear, many hours have passed, the toxin has been fully absorbed, it has already reached the liver and begun its work, and the window for any intervention that might prevent absorption has closed entirely. It is worth pausing here, in the soft middle of this explanation, to let the body settle a little more deeply into rest, to notice the weight of the arms and the heaviness of the eyelids, to let the voice wash over you like water that is just slightly warm, neither hot nor cold, simply present and steady and carrying you slowly along. There is nothing urgent here. The mushroom has been doing what it does for millions of years, and it will continue doing so long after this night is over, and all we are doing is observing it from the safe and comfortable distance of history and medicine, as one observes the behavior of some quiet and implacable force of nature, from a warm room with the rain falling outside. The second phase of Amanita phalloides poisoning begins when the silence ends, usually somewhere between 6 and 24 hours after ingestion, with onset of gastrointestinal symptoms that can be severe, and that typically include abdominal cramping, nausea, and persistent vomiting, together with significant disturbance of the digestive tract. These symptoms can be so pronounced as to cause dehydration, and they can mimic a range of other, less serious conditions, from ordinary food poisoning to viral gastroenteritis. And it is at this stage that misdiagnosis has historically been most likely because a physician presented with a patient suffering from vomiting and abdominal pain who does not know that mushrooms have been consumed or who is not told which mushrooms were consumed has little reason to suspect amatoxin poisoning from the symptoms alone. In earlier centuries, of course, the connection between the specific mushroom and the specific clinical course was not understood at all, and physicians working without any knowledge of toxicology as a discipline would have had essentially no framework within which to interpret what they were seeing other than the general observation that the patient had eaten mushrooms and was now unwell, a connection that the more observant among them did make, and which accumulated slowly over centuries into a body of folk knowledge that warned against certain species, often inaccurately and incompletely, but with a core of genuine experience at its center. The third phase is the one that determines the outcome, and it typically begins somewhere between 24 and 48 hours after ingestion, when the gastrointestinal symptoms may temporarily ease, creating a period of apparent improvement that physicians today recognize as deeply misleading, a calm that does not indicate recovery, but rather the moment when the focus of damage is shifting from the gastrointestinal tract to the liver and kidneys. During this phase, laboratory tests would reveal a sharp rise in liver enzymes in the blood, indicators of hepatocyte damage and death, and over the following days, the liver's function would decline progressively, leading to a condition known as acute liver failure, which carries with it a set of profound secondary consequences, including the accumulation of toxins in the blood that the damaged liver can no longer process, the impairment of coagulation because the liver can no longer produce the proteins required for normal blood clotting, and the development of a condition called hepatic encephalopathy, in which the accumulated metabolic waste products in the blood cross into the brain and produce a progressive alteration of consciousness that may begin as confusion or mild disorientation and progress through deepening stages of neurological impairment toward coma. Death, when it comes, typically occurs somewhere between 6 and 16 days after the mushroom was eaten, and it comes from this cumulative organ failure, from the quiet and methodical destruction that alpha amanitin has been carrying out in the body since the first hours after ingestion, working with a patience that seems, in the context of human suffering, almost incomprehensible. Approximately half of one death cap mushroom, roughly 30 grams of fresh tissue, contains enough amatoxin to kill a healthy adult human being, and the mushroom contains no warning, no bitterness, no burning sensation, no obvious signal of any kind to indicate that it is different from the many edible mushrooms that it closely resembles, particularly in its early button stage when the cap has not yet fully opened, and the distinctive features that distinguish it from edible species are least apparent. It is this resemblance, this quiet deception, this apparent innocence that has made the death cap so historically significant as an agent of both accidental and deliberate harm. And it is this quality that brings us now, having spent this first long chapter of our story, laying the groundwork of biology and chemistry that we will need for everything that follows, to the ancient world and to the first great human stories that have come down to us through the centuries, in which this pale and lovely mushroom moved from the forest floor into the kitchens and banquet halls of the most powerful people in the ancient world and changed the course of history in ways that we are still, in some respects, feeling today. Outside the window of wherever you are, the world is quiet now, or growing quieter, and the long night stretches ahead with its particular kind of generosity, its unhurried gift of hours. And we have many hours still, and many chapters still. And there is no need to hold on to the beginning when the middle is already here. And the end is very far away, and all the chapters between are soft and slow and full of the quiet details of the past. We return now to Rome, to the autumn of the year 54 of the Common Era, to a city that was at that moment the centre of the known world, a city of perhaps 1 million inhabitants, of marble and concrete and painted plaster, of aqueducts and sewers, of senators and slaves, of gladiatorial games and philosophical schools, of a political culture that combined extraordinary administrative sophistication with a degree of personal violence and dynastic intrigue that would seem remarkable even by the standards of later centuries. The emperor at this moment was Claudius Tiberius Claudius Caesar Augustus Germanicus, the fourth emperor of the Julio-Claudian dynasty, a man who had come to power in unusual and somewhat undignified circumstances in the year 41 of the Common Era, after the assassination of his nephew Caligula by members of the Praetorian Guard, who had dragged Claudius out from behind a curtain in the palace, where he had been hiding in terror, and proclaimed him emperor, apparently more from their own institutional interests than from any particular belief in his fitness for the role. Claudius had been born with a range of physical difficulties that had caused his own family to treat him as something of an embarrassment, keeping him away from public life and political roles throughout his youth and early adulthood, and the conventional wisdom of the Roman aristocracy had held him to be weak, unstable, possibly impaired in his reason, a figure of pity and mild contempt. The historical record, however, tells a more complicated story, because the emperor who emerged from behind that curtain and ruled Rome for the next 13 years turned out to be a genuinely capable administrator, a scholar of considerable learning who had written historical works on the Etruscans and the Carthaginians, an emperor who expanded the empire, reformed the legal system, completed significant public works, and governed with a degree of efficiency and competence that surprised those who had underestimated him. He was also a man who loved to eat, a man known for his appetites, who drank heavily and dined with enthusiasm, and who had a particular fondness for mushrooms, a fondness that, according to the accounts that have come down to us, would prove to be the detail on which the whole arc of his life eventually turned. By the autumn of 54 of the Common Era, Claudius was 63 years old and had been emperor for more than 13 years. His fourth wife, Agrippina the Younger, was a woman of formidable intelligence and ambition, the great-granddaughter of the first emperor Augustus, the mother of the young man who would become the Emperor Nero, and a political actor who had been maneuvering for years to secure the imperial succession for her son rather than for Claudius's own biological son, Britannicus, who was approaching the age at which he could formally be presented as a potential heir. The ancient sources, particularly the historian Cassius Dio, writing in the late 2nd and early 3rd centuries of the Common Era, and the biographer Suetonius, writing in the early 2nd century, and the historian Tacitus, whose account in the Annals is among the most detailed and most often cited, all record a version of events in which Agrippina, perceiving that Claudius was considering passing the succession to Britannicus, and thereby marginalizing her son Nero, decided to act first, and that she did so by arranging for poison to be introduced into a dish of mushrooms, served to the emperor at a banquet. Cassius Dio's account is notably specific, describing how Agrippina engaged the services of a woman named Locusta, who had recently been convicted of poisoning and who possessed an extensive practical knowledge of toxic substances, and how together they devised a preparation that was added to a particular mushroom in the dish of Caesar's mushrooms. Amanita caesarea, the prized edible species that was a delicacy of the Roman table, a mushroom that the emperor was known to enjoy, and that was similar enough in appearance to Amanita phalloides that the substitution or adulteration might easily go unnoticed by those preparing the meal. The account in Dio describes Claudius eating the poisoned mushroom, and then being carried from the banquet apparently overcome by wine, which was not an unusual occurrence. And then, during the night, the poison taking effect, and the emperor dying without having been able to speak or hear a word. On the 13th of October, in the year 54 of the Common Era, the debate among historians and physicians about the actual cause of Claudius's death has continued for many centuries and has not been fully resolved because the ancient sources, while unanimous in attributing the death to poison and in naming mushrooms as the vehicle, are not unanimous on every detail. And some modern scholars have raised questions about whether the clinical course described in the accounts is fully consistent with Amanita phalloides poisoning as we understand it today. The timing, for example, presents some difficulty because the death cap typically produces its most severe effects over a period of days rather than hours, and the death of Claudius, as described in some accounts, appears to have been relatively rapid, though Tacitus hints at a more prolonged course involving the intervention of a physician named Xenophon, who may have accelerated the end by other means, which is a detail that is itself debated. Scientific American published an analysis in February 2001, noting that some scholars believe muscarine rather than amatoxin may have been the relevant toxin, though the fungal source of muscarine presents its own complications. What is not in serious dispute among ancient historians is that Claudius died under circumstances that his contemporaries, including those who knew him and observed the court closely, attributed to deliberate mushroom poisoning, and what is also not in dispute is the consequence of that death. Because after Claudius died in October of the year 54, Agrippina's son Nero was immediately proclaimed emperor by the Praetorian Guard, and Britannicus died the following year, and Agrippina herself was murdered by her son in the year 59. And the reign of Nero unfolded over the next decade in ways that shaped the later Roman Empire so profoundly that historians of every subsequent era have returned to it again and again, tracing the long and winding consequences of that single autumn night, that single dish of mushrooms, that single quiet death. And Nero, it is said, was heard to remark with some dark humour that mushrooms were indeed the food of the gods, since it was through a mushroom that Claudius had been elevated to the divine status, that Roman tradition conferred upon its deceased emperors, and the remark was repeated and preserved, and has come down to us across 2,000 years as a kind of epitaph, half sardonic, half truthful, for a death that was both intimate and imperial, both personal and historical. There is something in the quality of the night that deepens around this hour, something in the way the body begins to release its hold on the concerns and movements of the day. And if you are still listening to these words, still following this voice down the long corridor of Roman history, then perhaps you can feel that deepening now, that softening of attention, that is not the loss of interest, but the loosening of the grip, the way a hand relaxed in sleep no longer holds what it held when awake. We are in no hurry here. The past is patient, and the night is long, and there are many more chapters and many more centuries to move through before we come to rest. And each of them will arrive in their own time, like stations on a slow night train through a countryside you cannot quite see in the dark, but can feel the movement of, the slight rocking, the distant rhythm of the rails, the occasional soft light of some unknown town passing by the window and then gone. And then the dark again, and then the next town, quiet and brief, and then behind you. After the death of Claudius, the court at Rome continued its intricate and often violent life, and mushrooms continued to be eaten at Roman banquets with the kind of enthusiasm that the culinary culture of the empire encouraged. And the death cap continued to grow in the oak forests outside the city, unhurried and indifferent to the fates it had or had not shaped, rising each autumn from its mycelium with the quiet regularity of a natural process that has no knowledge of courts or emperors or the long consequences of individual deaths. The reign of Nero ended in the year 68 of the Common Era with his suicide, and the empire passed through a year of civil war, and then settled into the reign of the Flavian dynasty. And the centuries turned one after another, slowly, and the empire grew and contracted, and eventually, in the West, gave way to something different, something that the people living through it did not yet have a name for, but that we have come to call the medieval period. And through all of those centuries, the death cap was present in the forests of Europe, continuing to fruit beneath its oak trees, continuing to offer its pale and beautiful cap to anyone who might pass by, continuing to hold its toxin with the deep patience of a thing that does not need to hurry because it has all the time in the world. We move now with the gentle and unhurried pace that this story requires, from the ancient world to a very different era, from the marble corridors of Rome to the drafty stone halls of medieval Europe, from an empire at the height of its power to a continent in the slow, complicated process of reorganizing itself after the fall, building new institutions and new power structures from the ruins of old ones, and finding, in the forests and fields and kitchen gardens of 1,000 small communities, both the sustenance it needed and sometimes the danger it did not see coming. The medieval period in Europe, stretching broadly from the 5th to the 15th centuries of the Common Era, was a time in which the knowledge of plants and fungi was held primarily by those we might call herbalists or apothecaries, or, in the monastic tradition, the learned monks who maintained the great libraries and gardens of the monasteries, and who often served their communities as physicians, surgeons, and pharmacists, all in one. The understanding of poisonous mushrooms in this period was an inherited and imperfect thing, passed down through a mixture of classical texts, folk tradition, practical experience, and occasionally superstition. And the boundary between factual observation and imaginative elaboration was not always clearly drawn. The writings of the ancient physicians, including Dioscorides and Galen, were copied and recopied in the scriptoria of monasteries across Europe, and the passages describing the dangers of certain mushrooms were transmitted through this chain of copying, sometimes with additions and sometimes with losses, into a medical culture that was doing its best to make sense of the natural world with the tools available to it. One of the most significant aspects of medieval fungal knowledge, and one worth dwelling on for a moment in the quiet of this hour, is the degree to which the distinction between edible and poisonous mushrooms was understood primarily through experience rather than through any systematic botanical or chemical analysis. There was no microscopy, no chemical analysis, no laboratory in any modern sense, and the methods by which medieval foragers and cooks tried to determine whether a mushroom was safe to eat were based almost entirely on accumulated folk wisdom, on the memory of what had been eaten safely before, and what had caused illness on the careful attention of experienced foragers who knew their local forests and could read the subtle distinctions between species that might look almost identical to the untrained eye. The deathcap, Amanita phalloides, presented particular difficulty in this regard because its resemblance to several edible species was not merely superficial but quite precise, particularly in its young button stage when it emerges from the ground, enclosed in its white universal veil, and presents very little of the distinctive coloration or form of the mature cap. And at this stage, it could easily be mistaken for any number of small, pale, edible mushrooms that grew in the same woodland environments. The Amanita caesarea, the Caesar's mushroom, which had been prized by the Romans and continued to be eaten in parts of southern Europe through the medieval period and into the present, shared certain features of habitat and appearance with the deathcap. And in regions where both species grew, the possibility of confusion was real and persistent, and the consequences of that confusion were recorded with sufficient regularity that the connection between certain mushrooms and certain deaths became part of the practical medical knowledge of the period, even without any theoretical framework to explain why the mushrooms caused the harm they caused. Medieval physicians working within the framework of Galenic humoral theory, which understood health and disease as the product of balance or imbalance among the four bodily humors, would have interpreted mushroom poisoning as a profound disruption of that balance, a severe and rapid imbalance of qualities that the body could not correct by its own regulatory processes. And the treatments proposed in medieval medical texts for mushroom poisoning reflect this theoretical framework, while also incorporating practical observations that, in retrospect, had some genuine value. The prescription of emetics to induce vomiting, for example, which appears in numerous medieval medical texts as a treatment for poisoning of various kinds, would have been genuinely useful in the very early stages of Amanita phalloides poisoning, before the toxin was fully absorbed, though by the time a patient was symptomatic, which, as we have noted, does not occur until 6 hours or more after ingestion, the window for effective gastrointestinal decontamination would in most cases have already closed, and the emetic would have been bringing up contents that no longer contained much of the toxin. The prescription of theriac, a complex polypharmaceutical preparation with roots in ancient Greek medicine that was used throughout the medieval period as a kind of universal antidote, would have had no specific effect on amatoxin poisoning, but might have provided some general supportive benefit, and it was administered with good faith by physicians who believed in its efficacy on the basis of a theoretical tradition that had no way of testing its claims systematically. The observation that certain patients recovered and others did not was attributed to variations in the constitution of the patients, to the degree of divine favor or disfavor, to the skill of the physician, and to the particular characteristics of the specific mushrooms eaten, rather than to any quantitative analysis of the dose of toxin received, which is the framework that modern toxicology would eventually bring to bear on precisely this question. It is in this medieval context that the history of the death cap begins to accumulate its second great historical event, a death that, like the death of Claudius, would have consequences far beyond the individual and the immediate, touching the lives of kingdoms and dynasties and wars, shaping the political map of a continent for generations to come. The Holy Roman Emperor Charles VI, who ruled from 1711 until his death in October of the year 1740, was, by all accounts, a man who enjoyed the pleasures of the table and who was known to appreciate the culinary traditions of the various regions of his vast and complicated empire, which stretched across a large portion of Central Europe and encompassed dozens of different cultural and linguistic communities. In the early autumn of 740, by the account that has been preserved in various historical sources and repeated in subsequent centuries, Charles sat down to a meal that included mushrooms that had been gathered with the intention of providing him with a delicacy, and that turned out to contain, whether through misidentification or some other misfortune, an amount of Amanita phalloides, sufficient to begin the process that we have already described in some detail. The silent absorption, the deceptive latency, the eventual clinical collapse. Within hours of the meal, Charles became deeply unwell, and his physicians were summoned, and they did what they could. Within the medical tradition of the 18th century, which was a tradition that was beginning to incorporate some of the early methods of what would become modern medicine, but which had not yet developed any specific treatment for amatoxin poisoning. Because the very concept of amatoxin did not yet exist, the chemical composition of the death cap would not be meaningfully analyzed for another century and a half. The emperor lingered for 10 days, according to the historical accounts, in increasing distress, and then died in Vienna in October of 1740. The death of Charles VI set in motion a cascade of events that historians have studied for nearly 3 centuries under the name of the War of the Austrian Succession. Charles had died without a male heir, and he had spent much of his reign attempting to secure the succession of his daughter, Maria Theresa, to his titles and territories through a legal instrument called the Pragmatic Sanction of 1713, which required the agreement of the other European powers to accept a female ruler, and which he had obtained from most of them through a combination of diplomacy, negotiation, and territorial concession. When he died, however, several of those powers immediately contested their agreements, most notably King Frederick II of Prussia, who within 2 months of Charles's death had invaded the Habsburg province of Silesia, triggering a war that would involve nearly every major European power. That would last until 1748. That would reshape the political boundaries of Central Europe, and that would introduce to the world a figure who would become one of the most significant military and political leaders of the 18th century, the Prussian king, who came to be known as Frederick the Great. The War of the Austrian Succession, with its hundreds of thousands of dead, its displaced populations, its rearranged maps and altered alliances, traces a long and indirect, but arguably traceable lineage back to an autumn meal in 1740, to a dish of mushrooms, to the quiet and patient work of a toxin that had been doing the same work in human bodies since long before the first European empire was founded, and the awareness of this lineage, even understood loosely, and with all appropriate historical caution, has a quality of strange and melancholy weight that settles slowly over the mind, like a fine layer of forest soil, settling over the root system of some great tree, building up year by year into the soft dark earth, from which the next generation of mushrooms will eventually emerge, as they always do, as they always have, indifferent to the wars and the kings and the long, complicated grief of history. The night is quieter now, perhaps, than it was when we began. And if you have been following this voice through Rome and through the medieval centuries and into the early modern period, through the courts of emperors and the chambers of physicians who did not yet know the name of the toxin they were trying to treat, then perhaps you have felt the hours passing in that particular way they pass, when the mind is occupied with distant things, with stories that are interesting enough to hold the attention loosely, but not urgently enough to keep it tense and awake. And perhaps the body has been settling through all of this, degree by imperceptible degree, into the comfortable geography of rest. We are going now to step forward from the death of Charles VI by almost 200 years, to move from the Age of Enlightenment, with its emerging scientific institutions and its growing appetite for systematic observation and rational explanation, toward the 19th century and the first genuine scientific investigations of the death cap and its toxins, investigations that would take place in the new laboratories of European universities, in the hands of chemists and pharmacologists and mycologists who were applying to the natural world a new set of tools and a new set of questions, and who were beginning, slowly and painstakingly, to crack open the chemistry of the most lethal mushroom in the world. But before we arrive at those laboratories, we should pause here. In the early 18th century, at a moment when the death cap's history is about to acquire one more royal victim, one more name to add to the list of the powerful and the unfortunate, in the form of a man whose death, unlike those of Claudius and Charles VI, would carry with it a specific irony that even the most serious historian might be permitted to appreciate, gently and without emphasis, as one appreciates a detail in an old and quiet painting that reveals itself only after long looking. Pope Clement VII, born Giulio di Giuliano de' Medici in Florence in 1478, was the son of Giuliano de' Medici, who had been assassinated in the Pazzi conspiracy of 1478, and a member of one of the most powerful and culturally significant families in the history of Florence and of the Italian Renaissance more broadly. He became pope in November of 1523 and served in that role until his death in September of 1534, a pontificate of 11 years, during which he navigated one of the most difficult periods in the history of the Roman Catholic Church, presiding over the years of the Protestant Reformation, the catastrophic sack of Rome in 1527, and the contentious diplomatic crisis surrounding the English king, Henry VI's request for an annulment of his marriage to Catherine of Aragon, a request that Clement famously declined to grant, a decision that would lead directly to the English Reformation and the establishment of the Church of England. Another of those pivotal moments in European history whose distant reverberations are still felt today. The Pope died on the 25th of September 1534 in Rome, and the circumstances of his death were the subject of rumour and speculation among his contemporaries and among later historians, with some accounts attributing his death to the consumption of death cap mushrooms, either accidentally in the ordinary course of a meal or deliberately as an act of poisoning. The historical consensus, as carefully assembled by scholars including Louis Cilliers and F.P. Retief in their study of diseases and causes of death among the popes, tends toward natural causes, specifically the accumulated physical and psychological toll of an extraordinarily difficult pontificate. But the rumour of mushroom poisoning circulated widely enough and persistently enough to earn its place in the longer history of the death cap as one of those historical associations that may not be firmly established, but that reflects the degree to which this particular mushroom occupied the imagination of European culture as an instrument of both accidental tragedy and deliberate violence. It is worth noting that Voltaire, in a later century, would add his own voice to the speculation around Clement's death, claiming the death cap as the cause, and while Voltaire was a man of considerable literary and philosophical gifts, he was not always a man of strict historical precision, and his attribution of the death to mushroom poisoning should perhaps be held with some of the gentle skepticism that one extends to colourful details. That one would like to be true. There is something rather beautiful, in a quiet and unhurried way, about the persistence with which the death cap appears at these hinge points of European history, at the deaths of emperors and popes, and the origins of wars, not because the mushroom seeks out the powerful, but simply because the powerful were also human, also subject to the ordinary hungers and pleasures of embodied life, also sitting down to meals and reaching for dishes of food prepared by others, also moving through a world in which the distance between a delicious dinner and a fatal one could be measured not in any visible quality of the thing consumed, but only in its chemical composition, invisible to the eye, indistinguishable to the taste, waiting in the quiet of the forest floor for the season to fruit, and then fruiting, as it has always fruited, without intent, without malice, without any of the qualities we might attribute to it, if we were not careful to remember that it is a mushroom, not an actor, not a conspirator, not a force of history in any purposive sense, only a living thing doing what living things do, which is to persist through time, to reproduce, to follow the patterns laid down in its chemistry and its biology, and to allow the rest of us to draw from it whatever meaning we find there in the long and quiet hours of a dark and restful night. Meanwhile, in the laboratories and botanical gardens and academic institutions of 19th century Europe, a different kind of encounter with the death cap was beginning to take shape, one conducted not over a dinner table, but over a microscope and a chemical apparatus, by men who asked not what the mushroom had done to the famous dead, but what it was actually made of, what compounds it contained, how those compounds worked in the tissues of the body, and whether any intervention might be found to interrupt their work before it reached its conclusion. And it is to that story, that long and careful and still unfinished story of science, facing one of nature's most elegant poisons, that we will turn our attention in the chapters that follow, moving as we always move in this story, gently, without urgency, letting each detail arrive in its own time, like the slow return of light at the end of a long winter night. The early scientific study of toxic mushrooms in Europe unfolded across the 19th century in the context of a broader flowering of natural history and chemistry that was transforming the understanding of the biological world, a pace that had no real precedent in earlier human experience. The mycologist Elias Magnus Fries, the Swedish botanist who is often credited with laying the systematic foundations of modern mycology, published his monumental work Systema Mycologicum between 1821 and 1832, and within this vast and methodical catalogue of fungal species he described, and formally named Amanita phalloides, with the precision and care of a man who understood that accurate identification was the first prerequisite for any further scientific work, including the work of understanding which species were dangerous and why. The formal scientific naming of the death cap was itself a small but significant step in the longer history of efforts to understand and address the dangers it presented, because before a thing can be studied systematically, it must first be named, distinguished from other things, given a fixed identity in the scientific record that allows observations from different researchers in different places to be reliably compared with one another. And Fries's taxonomic work provide that foundation not only for the death cap, but for the hundreds of other fungal species that he catalogued in his lifetime, establishing a framework within which mycology could develop as a coherent scientific discipline rather than a collection of disconnected local observations. During the same period, The discipline of organic chemistry was undergoing its own revolutionary development, as chemists in Germany and France and Britain were developing new techniques for isolating and characterizing the chemical components of natural substances, techniques that would eventually be applied to the toxins of the death cap, though the application would come slowly and with considerable difficulty because the amatoxins are complex molecules whose isolation and characterization required advances in chemistry that were not yet available in the early part of the 19th century. The first reasonably systematic attempt to describe the clinical effects of Amanita phalloides poisoning in terms that would be recognizable to modern medicine was made by a number of European physicians working in the mid to late 19th century, as both mycology and clinical medicine were becoming more rigorous disciplines with clearer standards of evidence and more careful methods of observation. French physicians and toxicologists contributed significantly to this early literature, as France had a long tradition of both culinary mycology, the enthusiastic collection and consumption of wild mushrooms, as a significant element of the national food culture, and correspondingly, a significant burden of mushroom poisoning cases that provided the clinical material from which generalizations could be drawn. The pattern of delayed symptom onset, following a period of apparent good health, the progression from gastrointestinal symptoms to liver involvement, the characteristic course of decline over several days. All of these features were observed and described with increasing accuracy as the century progressed. And by the final decades of the 19th century, the clinical picture of severe Amanita phalloides poisoning was reasonably well understood by the physicians most experienced with it, even though the underlying biochemical mechanisms remained entirely unknown, and treatment remained essentially supportive, a matter of providing comfort and managing complications, while the body did or did not manage to survive the toxin on its own resources. It is worth noting, with the reflective quietness this story invites, that this period of improving clinical knowledge coincided with a period of enormous confidence in the capacity of science and medicine to eventually explain and solve the problems that nature presented, a confidence that was in many ways justified by the genuine advances that were being made. But that also contained within it a certain optimism that would be tested by the death cap, among other things. Because the gap between understanding a problem and solving it has always been wider than optimism alone would suggest. And the death cap would continue to kill people throughout the 20th century and into the 21st, even as understanding of its mechanism grew progressively more precise and detailed. It is appropriate to pause here at the end of this long and slowly moving survey of the mushroom's early history, to take a breath, to feel the chest rise and fall, to notice how the body has travelled through this hour of history without going anywhere at all, lying still in the dark, while ancient Rome and medieval Europe and the courts of emperors have passed through the mind like shadows on a wall, present and then gone, and leaving only a faint warmth behind, the residual warmth of stories encountered and then released. The death cap does not know that it is infamous. It does not know that it has been named in the chronicles of Roman emperors, in the speculation of historians and doctors, in the quiet wonder of naturalists, who held its pale cap in their hands and tried to understand how something so small and so beautiful could contain such consequences. It grows because it is its nature to grow, beneath the oak trees of Europe, and now on other continents too. And it fruits each autumn because that is what it does, and it holds its toxin not as a weapon but as a biochemistry, and the harm it causes is incidental to its existence in the most literal sense, a side effect of a chemistry evolved for purposes that have nothing to do with human beings at all, purposes that are about mycelium and spores and the slow negotiation of life in the soil, purposes so remote from human affairs that to regard the mushroom as malevolent is a kind of category error, like being angry at the rain. And yet the rain can flood, and the mushroom can kill, and the distance between the forest and the table has always been shorter than it might seem, and the history of that short and sometimes fatal distance is a history worth knowing, worth holding gently in the mind on a quiet night, as a reminder of the many ways in which the natural world is simultaneously indifferent and intimate, both completely other and completely present, and as present now as it has ever been, in the forests outside whatever window you have, in the soil beneath whatever ground you walk on, patient and unhurried and old beyond imagining, while you rest here, quietly, and let the night do what the night does best. And so we arrive with the same unhurried pace that has carried us through Rome and through the medieval centuries, and through the courts of emperors whose deaths echo still in the long corridors of historical consequence, at the 19th century proper, at a period when the tools available to those who wished to understand the death cap were beginning, slowly, and with great effort, to become adequate to the task, and when the first tentative steps were being taken toward a chemistry that could name not just the mushroom, but the specific molecules within it that carried its lethal power. It is a period worth dwelling in for some time, because it is the period in which the story of the death cap shifts in character, moves from the domain of narrative and chronicle into the domain of experiment and measurement, from the world of diaries and court records into the world of laboratory notebooks and published papers in the emerging scientific journals of the age, from a story told about what happened to a story told about why it happened and how, and this shift, gradual and incomplete as it was, represents one of the genuinely significant transitions in the long relationship between human beings and this particular mushroom. The 19th century in Europe was a time of extraordinary intellectual energy, a time when the methods of natural science were being applied with increasing confidence to every domain of the natural world, from astronomy to geology to biology to chemistry, and when the boundaries between these disciplines were permeable enough that a single curious mind could move between them without great difficulty, carrying observations from one field into another, and finding connections that a more rigidly specialized culture might have missed. It was in this environment of wide-ranging scientific curiosity that the systematic study of poisonous fungi found its footing in the work of men who were simultaneously naturalists, chemists, physicians, and collectors who went into the forests with their notebooks and their specimen jars, and brought back what they found to be subjected to the careful attention of the new sciences. The German chemist Justus von Liebig, one of the founding figures of organic chemistry, had by the middle of the century helped establish the analytical techniques that made it possible in principle to isolate and characterize the chemical components of complex organic substances. And while Liebig himself did not work extensively on fungal toxins, the tools and methods he developed and taught were the tools that subsequent researchers would eventually apply to the problem of what exactly was inside a death cap mushroom that made it so reliably and so profoundly deadly. Meanwhile, in France, the physician Pierre Victor Renaud was publishing careful clinical descriptions of mushroom poisoning cases in the 1840s, documenting with unusual precision the sequence of symptoms, and the typical course of decline, and laying a clinical foundation that later physicians and toxicologists would build upon, adding detail and nuance as their own methods improved. In Germany, the botanist Heinrich Bernhard Rupp had contributed to earlier mycological descriptions, and the tradition of careful botanical observation that ran from Linnaeus through Fries, through the many naturalists of the 19th century, was producing an increasingly reliable picture of fungal diversity, a picture in which the death cap occupied a prominent and precisely delineated place. The pace of this scientific work was slow, measured in decades rather than years, and the death cap continued to claim lives throughout the century, with perfect indifference to the progress being made in the laboratories. And botanical gardens, as it has always done, because the gap between scientific understanding and clinical application has never been a small one, and the amatoxins were not molecules that yielded their secrets easily to the methods available before the 20th century. By the end of the 19th century, the clinical description of Amanita phalloides poisoning was reasonably complete. The mushroom itself was well characterized in the mycological literature, and the connection between the species and its deadly effects was firmly established. But the specific chemical identity of the toxins remained unknown, and treatment remained entirely supportive, and the death toll continued to accumulate in the hospitals and homes of Europe. And wherever European immigrants had carried the mushroom, knowingly or not, to new continents. And so the 19th century drew to its close with the death cap standing at the threshold of a new era of science, not yet fully understood, not yet susceptible to any specific medical intervention, but increasingly visible in the crosshairs of a scientific culture that was beginning to develop the tools it would need to look more closely. The transition from the 19th to the 20th century brought with it a remarkable acceleration in the pace of chemical and biological research, an acceleration driven in part by new technologies, in part by new institutional structures that supported scientific work on a larger and more coordinated scale, and in part by the sheer accumulation of knowledge and technique that made each new advance possible in ways it had not been a generation earlier. It was in this context that the first serious attempts to chemically characterize the toxins of Amanita phalloides were made, attempts that would span several decades and involve the work of researchers across multiple countries and disciplines, and that would ultimately reveal a molecular architecture of considerable elegance and considerable complexity. The German chemist Heinrich Wieland, who would later win the Nobel Prize in Chemistry in 1927 for his work on bile acids, was among the early researchers who turned their attention to fungal toxins in the early decades of the 20th century, and his work and the work of his colleagues began to establish the chemical framework within which the amatoxins would eventually be fully characterized. The research was painstaking and required enormous quantities of mushroom material because the toxins were present in relatively small concentrations in the fresh tissue, and extracting and purifying sufficient quantities for chemical analysis demanded both patience and a reliable supply of the mushrooms themselves, which had to be collected from the wild, since no one had yet managed to cultivate Amanita phalloides under laboratory conditions, a feat that remains essentially impossible today because of the obligate mycorrhizal relationship between the death cap and its host trees, a relationship so intimate and so specific that the fungus cannot complete its life cycle without the living roots of a compatible tree. It is a particular irony of the death cap's chemistry that the very specificity and complexity of its toxins, the qualities that make them so interesting to chemists and so dangerous to the people who consume them, are also the qualities that make them so difficult to study. Because purifying complex cyclic peptides from biological tissue using the methods available in the early 20th century was a genuinely difficult undertaking, prone to degradation and contamination, and loss of material at every step. Despite these difficulties, progress was made, and by the late 1930s and early 1940s, researchers including Fyodor Leinen, who would himself later win a Nobel Prize for his work on fatty acid metabolism, and his colleagues in Germany were making significant advances in the isolation and partial characterization of the toxic components of the death cap, advances that were published in the German chemical literature. And that laid important groundwork for the more complete characterization that would come after the Second World War. The war itself interrupted scientific progress across Europe in ways that were both dramatic and deeply consequential for individual researchers and for whole institutions. And the work on amatoxin chemistry, like so much other scientific work, was slowed, and in some cases halted entirely by the disruptions of the conflict, only to resume in the post-war period with new urgency and new resources. The post-war period in Germany and in European science more broadly was a time of reconstruction and reinvention, when scientific institutions were being rebuilt and when international scientific communication was being restored after years of isolation. And it was in this environment that the most significant advances in the chemistry of the death cap toxins would eventually take place in the hands of a chemist whose name deserves to be held in the memory of anyone who has followed this story with serious attention. That chemist was Theodor Wieland, the son of Heinrich Wieland, and he would dedicate a significant portion of his scientific career to the systematic characterization of the amatoxins and phallotoxins of Amanita, phalloides, work that he carried out primarily at the University of Frankfurt, and that continued from the post-war years, well into the latter decades of the 20th century. Theodor Wieland's work on the structure of the amatoxins was meticulous and methodologically innovative, requiring the development of new chromatographic techniques for the separation of closely related cyclic peptides, and it resulted in the complete structural characterization of alpha-amanitin and the other amatoxin variants, establishing definitively that they were bicyclic octopeptides, ring-shaped molecules of 8 amino acids, connected in a specific spatial arrangement that was unlike anything previously described in the chemical literature. The determination of these structures was published in a series of papers in the 1950s and 1960s, and each publication added a layer of precision to the understanding of what the death cap actually contained and how its toxins differed from one another in subtle but important ways, with alpha-amanitin emerging clearly as the dominant lethal agent and beta-amanitin, and the other variants playing secondary roles in the overall toxicological picture. The work of Theodor Wieland and his collaborators, including his long-standing association with the Italian-American biochemist Silvio Folstick, who contributed crucially to understanding the mechanisms by which the amatoxins reached their cellular target, represents a sustained and extraordinarily productive scientific partnership that produced insights not only relevant to mushroom poisoning, but to the fundamental biology of gene expression. Because the discovery that alpha-amanitin inhibited RNA polymerase II with exquisite specificity made it a powerful tool for cell biologists studying transcription. And it has been used in this way in thousands of experiments in the decades since its mechanism was elucidated. It is one of the more remarkable aspects of this molecule's history that a compound isolated from the world's most lethal mushroom should become a standard reagent in molecular biology laboratories around the world. A small brown bottle on a shelf in a university biochemistry department, carrying within it the same chemistry that killed emperors and popes, now deployed in the service of understanding how all living cells read their own genetic instructions. The structural work on the amatoxins was followed by equally important work on their synthesis, as Wieland and others worked to produce the compounds in the laboratory, without relying on the mushroom as a source, and the total chemical synthesis of alpha-amanitin was eventually achieved, a technical accomplishment of considerable complexity that confirmed the correctness of the proposed structure and opened new possibilities for studying the molecule and its effects in controlled experimental conditions. Meanwhile, the clinical understanding of amatoxin poisoning was advancing in parallel as physicians in Europe and elsewhere accumulated experience with the condition and published increasingly detailed case series and clinical studies that refined the understanding of the natural history of the poisoning and the factors that influenced its outcome. By the 1970s and 1980s, the basic framework of modern understanding of Amanita phalloides poisoning was in place, including the identification of RNA polymerase II, inhibition as the primary mechanism, the characterization of the three-phase clinical course, and the recognition that early diagnosis and aggressive supportive care were the most important determinants of outcome in the absence of any specific antidote. And it was in this same period that the question of treatment began to attract serious clinical research attention, with several European centres publishing studies of various proposed interventions. From high-dose penicillin to silibinin, a compound derived from the milk thistle plant Silybum marianum, that appeared in some cases to reduce the severity of liver injury when administered early enough in the clinical course. Silibin, derived from the seeds of the milk thistle, represents one of the more intriguing chapters in the treatment history of death cap poisoning, a chapter that unfolds at the intersection of traditional herbal medicine and modern pharmacology, in the way that several of the more interesting stories in medical history do, where the accumulated empirical wisdom of folk healers and herbalists turns out to contain a kernel of genuine pharmacological reality that only becomes fully visible when examined through the lens of modern science. Milk thistle had been used in European herbal medicine for centuries as a treatment for liver ailments of various kinds, an application that had been passed down through the herbal tradition from classical antiquity, appearing in the works of Dioscorides, and later in the great herbals of the Renaissance period, including the work of Leonhard Fuchs and the influential German herbal tradition, and the connection between milk thistle and the liver, was established in folk medicine long before anyone had the biochemical tools to understand what silymarin, the active complex of compounds in the plant, was actually doing at the cellular level. In the 1960s and 1970s, German pharmacologists began to investigate the chemistry and pharmacology of milk thistle more rigorously, isolating the specific compounds within it and testing their effects in experimental models of liver injury. And this work led to the purification and characterization of silibin and the other silymarins, and to the publication of studies suggesting that these compounds could protect liver cells against a range of toxic insults, including the damage produced by amatoxin. The proposed mechanism of silibinin's protective effect against amatoxin poisoning involves its ability to compete with alpha-amanitin for uptake into liver cells by the same transport proteins that the toxin uses to gain entry, so that if silibinin is present in sufficient concentration in the blood at the time that alpha-amanitin is being transported into hepatocytes, it can block that transport and reduce the amount of toxin that reaches its intracellular target. And this mechanism, if correct, would explain why silibinin has a meaningful window of opportunity in the early hours after ingestion and loses much of its utility once the toxin has already been taken up. Clinical case reports and small series from European centres, particularly in Germany, France, and Austria, began to accumulate in the 1970s and 1980s, suggesting that patients treated with intravenous silibinin within 24 to 48 hours of ingestion had better outcomes than those treated later or not at all. And by the 1990s, silibinin had become a standard component of the treatment protocol for serious Amanita phalloides poisoning in many European countries, administered intravenously in high doses alongside aggressive supportive care. The compound was not without complications, and it was not a cure in any simple sense, because its efficacy depended heavily on the timing of administration relative to the ingestion, and patients who presented late, as many did because of the deceptive latency of the poisoning, could not always be reached by the intervention before significant hepatocyte damage had already occurred. And in those cases, the supportive measures that remained available were insufficient to prevent progression to severe liver failure in a substantial proportion of patients. The story of silibinin is, in its quiet way, a story about the relationship between traditional knowledge and modern science, about the value of attending carefully to what generations of healers observed and recorded, even when those observations were embedded in theoretical frameworks that modern medicine no longer accepts, because the theoretical framework that led herbalists to associate milk thistle with liver protection was not correct in any modern biochemical sense. But the observation that the plant had a beneficial effect on certain kinds of liver injury turned out to be correct, and that correctness survived the abandonment of the theory that had originally generated it, and found a new home in the biochemistry of transport, proteins, and cyclic peptides. It is worth noting in the reflective register that this hour invites that this kind of survival of useful practical knowledge through the replacement of theoretical frameworks is not unique to the history of milk, thistle, or of mushroom poisoning, but is rather a general feature of the history of medicine, one that argues for a certain humility in the face of accumulated empirical experience, even when that experience cannot yet be explained in terms that the current theoretical vocabulary would recognize. Meanwhile, the development of liver transplantation as a viable clinical intervention in the 1980s and 1990s would add a new dimension to the treatment of the most severe cases of Amanita phalloides poisoning, offering a last resort for patients whose livers had failed beyond the capacity for natural recovery, and the first reports of successful liver transplantation for mushroom poisoning began to appear in the medical literature in the late 1980s, representing a remarkable, if extreme, application of one of the most complex operations in all of surgery to a condition caused by half a mushroom eaten in a moment of mistaken identification. And so the treatment of death cap poisoning moved through the 20th century from essentially nothing, from supportive care and hope, and the occasional application of interventions that had no specific scientific basis, toward a more organized and evidence-based approach. though always with the knowledge that the best treatment remained, as it had always been, not eating the mushroom in the first place. The geography of the death cap is a subject that has attracted increasing scientific attention as the 20th century gave way to the 21st, and for a reason that would have been entirely unexpected to the naturalists of the 19th century who first catalogued the species in the forests of Europe, a reason that carries with it some of the quality of a slow-moving ecological story, the kind of story that unfolds not over years or decades, but over generations, visible only to those who take the long view. Amanita phalloides is not a native species of North America, or of Australia, or of South Africa, or of South America, or of New Zealand, or of Japan, or of many of the other parts of the world, where it is now found growing in parks and gardens and wild lands, far from the European oak forests where it evolved, and where it has been part of the ecological community for millions of years. The mushroom arrived in these places through the movement of plants, specifically through the importation of European trees, particularly oak and cork, oak and chestnut, and other mycorrhizal tree species, which were brought to new parts of the world for timber, for amenity planting in gardens and parks, and in the case of cork oak, for the commercial production of cork, and which carried with them, in the soil around their roots, the mycelium of the fungi with which they had established their mycorrhizal partnerships, in Europe including, in many documented cases, the mycelium of Amanita phalloides. The death cap thus followed European colonialism and European horticulture around the world, arriving in new continents, not in the hands of settlers who brought it deliberately, but in the root systems of trees that were brought for entirely different purposes, establishing itself in new soils and new climates with a mycological efficiency that reflected the adaptability of an organism that had been making mycorrhizal partnerships with trees for millions of years, and was perfectly capable of doing so with the same tree species growing in California or South Africa or Southern Australia. The arrival of the death cap in North America, documented through mycological survey work and specimen collections dating to the late 19th and early 20th centuries, is associated with the planting of European oaks in parks and estates on the East Coast, and subsequently the spread of the mushroom westward as the century progressed, following the distribution of European trees planted in parks and gardens, and along streets, in cities and towns across the continent, until by the late 20th century, the death cap was well established along both coasts and in many interior regions as well. In California, where European oaks and other compatible tree species were widely planted throughout the 20th century, the death cap found particularly favorable conditions, and by the early 21st century, it had become the dominant cause of serious mushroom poisoning in the state, accounting for cases that disproportionately affected immigrant communities from East and Southeast Asia, particularly from China, Vietnam, and Laos, where foraging traditions involving edible mushrooms of similar appearance were a cultural practice carried to the new country, and where the visual familiarity of the death cap with certain prized edible Asian species, particularly the paddy straw mushroom, Volvariella volvacea, created a pattern of tragic misidentification that has recurred with painful regularity in California and in other parts of the United States and Australia, where similar demographic and ecological factors converge. The California Department of Public Health and the California Poison Control System have documented numerous cases of serious Amanita phalloides poisoning over the decades, including a particularly tragic cluster of cases in 1988 in Santa Cruz County, involving members of the Hmong community, in which a group of people foraged what they believed to be edible mushrooms from a local park and prepared and ate them in a traditional way, with multiple severe poisonings resulting, several requiring intensive medical care, and a pattern that would repeat itself in similar form in subsequent years and in other California counties, prompting public health campaigns and community outreach efforts aimed at the immigrant communities most at risk, efforts that have had some success, but that have faced the inherent difficulty of reaching diverse communities with diverse languages, diverse cultural practices around food and foraging, and diverse levels of engagement with public health systems. The story of the death cap spread and its disproportionate impact on immigrant communities in the late 20th and early 21st centuries is a story that carries within it a particular kind of social and cultural weight, the weight of displacement and unfamiliarity, of the deep human impulse to maintain connection with a homeland through the practices and foods of that homeland, of the way that a knowledge of the edible fungi of one's native forest can become, in an alien forest, a source of danger rather than sustenance, and of the complex responsibilities that public health systems bear when they serve populations whose lives have been shaped by experiences very different from those the systems were designed for. And in January 2026, a superb bloom of death caps in California following early fall rains caused a new cluster of poisonings, including deaths in Sonoma County, a reminder that the mushroom's work continues with absolute consistency in the present day, as it has continued through every century of the history we have been quietly tracing through this long and patient night. We turn now, as we must, to Australia, to a country that received the death cap in much the same way that California did, through the importation of European trees, and where the mushroom's establishment in the landscape has followed a trajectory that Australian mycologists and public health authorities have been tracking with increasing concern through the latter decades of the 20th century and the opening decades of the 21st. The death cap arrived in Australia almost certainly through the importation of European oak trees, Quercus robur and related species, which were planted extensively in the gardens and parks of the Australian Capital Territory, New South Wales, and Victoria from the late 19th century onward, as European settlers sought to create in the southern hemisphere landscape some echo of the northern European environments they had left behind, a horticultural impulse that was in many ways an expression of the broader colonial project of transforming the new land in the image of the old. And that had consequences, in this particular case, that no one at the time anticipated. The first documented death from Amanita phalloides poisoning in Australia was recorded in 1912 in Canberra, and subsequent decades produced additional cases. But it was the rapid expansion of the mushrooms range in the Australian Capital Territory from the 1970s onward that brought it to the sustained attention of Australian public health authorities and mycologists, as the death cap was found fruiting not only in established parks and gardens, but increasingly in suburban neighborhoods, where European oaks had been planted as street trees, bringing it into close proximity with residential areas, and with the same communities of Asian immigrants who were, as in California, at elevated risk because of the resemblance of the death cap to edible species familiar from their countries of origin. The Royal Botanic Gardens, Victoria, has maintained records of death cap specimens and poisoning cases, and has been involved in public education efforts around the mushroom and the work of mycologists associated with Australian botanical institutions has contributed significantly to the understanding of the death cap's ecology and distribution in the Australian context, including documentation of the ways in which the mushroom has continued to spread as its host trees have grown, and as the mycelium has extended through the soil networks of parks and suburbs. In the summer of 2023, Australia experienced one of the most widely reported death cap poisoning incidents in its history, when 3 members of a family died after consuming mushrooms that had been foraged in the Canberra region and prepared in a meal served at a family gathering, a tragedy that drew national attention in Australia, and that reignited public and policy discussion about the dangers of wild mushroom foraging and the particular risks faced by communities for whom foraging is both a practical for food tradition and a cultural one. The details of the 2023 Australian case, as they emerged through the investigation and the subsequent coronial inquiry, illustrated with particular clarity the way in which the death cap's deceptiveness operates not just at the level of individual sensory experience, the appearance, the smell, the taste that give no warning, but also at the level of cultural knowledge, where the expertise in wild mushroom identification that is entirely appropriate and reliable in one geographical and ecological context becomes dangerously inadequate in another, because the mushrooms of Europe and of Asia do not look exactly like each other and do not grow in exactly the same places, but they look similar enough and grow in similar enough places to create the conditions for exactly the kind of terrible mistake that ended in 3 deaths in Canberra in 2023. The coronial inquiry that followed the 2023 Australian deaths examined in careful detail the public health infrastructure around wild mushroom poisoning in Australia, the adequacy of warning systems and public education campaigns, the accessibility of information in the languages spoken by the communities most at risk, and the speed and effectiveness of the clinical response once the victims were identified as having consumed potentially toxic mushrooms, and the findings of the inquiry produced a set of recommendations that touched on each of these areas, calling for improved signage in parks and reserves where death caps were known to grow, enhanced multilingual public education materials, better training for emergency department clinicians, in the recognition and early management of amatoxin poisoning, and increased availability of psilocybin and other treatment resources at hospitals likely to receive such cases. It is worth reflecting, as we move through these details of recent Australian history, that every one of the clinical and public health challenges identified by the 2023 inquiry had been identified in earlier reports, in the reports following the 1988 California cases, in the European literature stretching back through the 20th century, and that the persistence of the problem, despite decades of awareness, reflects not any failure of knowledge or intention, but the genuine difficulty of reaching all the people who need to be reached with information that is specific enough to be useful and accessible enough to be heard, a difficulty that is not unique to mushroom poisoning, but that is particularly acute in this case, because the risk is so specific, so particular, so tied to a single moment of decision in a forest or a park that may occur only once in a lifetime. And over all of this, through all of these decades and all of these cases, the death cap itself has remained unchanged, growing as it has always grown, fruiting as it has always fruited, holding its toxin with the perfect equanimity of a thing that has no awareness of the attention being paid to it, no knowledge of the laboratories and the hospitals and the inquiries and the public health campaigns that circle around it. No sense of itself as anything other than what it is: a mushroom, a living thing, patient and complete. There is a quality to the middle of a night like this, a quality that is different from the early evening and different from the deep night that comes later, a quality of settled darkness, of time that has found its rhythm. And if you have been following this voice through the chemistry and the geography and the recent history of the death cap, through California and Australia, and the slow movement of a European mushroom across continents, it was never meant to inhabit, then perhaps you can feel that quality now. The way the room seems to have grown a degree or two more still, the way the body has given up a little more of its daytime tension, the way the mind has begun to hold the story more loosely, less as a sequence of facts to be retained, and more as a current to be carried by. We move now, in this quieter register, to a dimension of the death cap's history that we have not yet fully explored, a dimension that is neither purely scientific nor purely political, but sits at the intersection of culture and imagination, the dimension of the mushroom in human art and literature and mythology, in the stories that cultures tell themselves about the dangerous and beautiful things that grow in the world outside human settlement, just beyond the cultivated land, in the wild spaces where the rules of the domestic world do not fully apply. The relationship between mushrooms and human imagination is an ancient and multifaceted one, predating by thousands of years any systematic botanical or chemical understanding of fungi, and it encompasses both a deep fascination and a deep ambivalence. A sense of mushrooms as occupying a liminal space between the safe and the dangerous, the nourishing and the poisonous, the familiar and the utterly strange, and the death cap, with its elegant beauty and its catastrophic consequences, has always embodied this ambivalence more completely than almost any other species in the kingdom of fungi. The ancient Romans, who enjoyed mushrooms as a luxury food, and who were also aware of the fatal possibilities of certain species, developed a range of folk methods for testing whether a mushroom was safe, methods that were almost entirely ineffective, but that reflect the anxiety that accompanied the pleasure of mushroom eating, the awareness that the same category of object that could provide such pleasure could also, in a version almost indistinguishable from the pleasurable one, provide only suffering and death. The practice of cooking mushrooms with a silver spoon, in the belief that the silver would tarnish in the presence of a poisonous species, was recorded in Roman sources and persisted into the medieval period, and beyond, repeated in household manuals and cookery books as recently as the 19th century. Despite being entirely without foundation in chemistry, a piece of sympathetic magic dressed in the clothing of practical wisdom. The practice of throwing mushrooms away, if they turned black on cooking, or if they were noticeably bitter, or if they grew in particular locations, near trees or bodies of water that were considered inauspicious, reflected genuine attempts to identify patterns in a confusing and potentially lethal landscape, and some of these folk rules had genuine validity, identifying real correlations between certain visual features and certain toxic species, while others were pure superstition, and the difficulty lay in knowing which was which without a systematic science of mycology to distinguish between them. In the literature of the medieval and Renaissance periods, mushrooms appear repeatedly as symbols of the deceptive and the treacherous, of the beautiful thing that conceals a deadly interior, of the world's tendency to offer pleasure and danger in forms so similar as to be barely distinguishable. And these literary associations reflect and reinforce the cultural anxiety around mushroom eating. that was a genuine feature of the lives of people who gathered food from the wild and who had in their communities the living memory of deaths that had followed from the wrong choice in the forest. Shakespeare, writing at the end of the 16th century, refers to mushrooms in The Tempest in a way that aligns them with the supernatural and the uncanny, associating them with the circles of fairy rings and with the midnight work of spirits, a literary association that draws on a folk tradition of regarding mushrooms as things that appeared suddenly and mysteriously from nowhere, growing overnight in circles that seemed too perfectly geometric to be accidental, and that had therefore long been woven into the fabric of folk belief about the hidden agencies at work in the natural world. And the death cap, the most beautiful and most deadly of the European mushrooms, the one that looked most like an edible species, and tasted most like one, and gave no warning of its true nature until it was too late, occupied a special place in this cultural imagination as the embodiment of natural deception, the thing that seemed to be one thing and was entirely another, a symbol that resonated far beyond its literal botanical identity, into the broader human experience of encountering the world's treachery in a form so beautiful as to seem almost deliberate. The 20th century brought to the cultural history of the death cap a new set of dimensions that arose from the intersection of the mushroom's biology with the technologies and anxieties of a particular historical moment, specifically the moment in the late Cold War period, when the idea of biological weapons and natural toxins as potential instruments of state violence was being actively explored by intelligence services and military research programs on both sides of the geopolitical divide. The amatoxins of Amanita phalloides, with their extraordinary potency, their chemical stability, their colorlessness and tastelessness and odorlessness, and their delayed onset of action, possessed in theory many of the characteristics that would be valued in a covert poison, and it is therefore not entirely surprising that they attracted some attention in the context of state-sponsored research into toxic agents, though the details of this research, to the extent that it existed, remain largely classified or otherwise inaccessible to the historical record. What is more accessible is the history of the amatoxins in the context of targeted killings attributed to intelligence services, a history that centers most prominently on a compound related to the amatoxins called amanitin, conjugated to monoclonal antibodies, which has been explored in cancer therapy research, and on the broader question of natural toxins as assassination tools, a question that became practically rather than theoretically relevant in a number of documented and alleged cases in the latter half of the 20th century. The development by researchers at the Max Planck Institute and elsewhere of amatoxins antibody conjugates as experimental cancer treatments, beginning in the 1990s and continuing into the 21st century, represents a remarkable reversal of the mushroom's usual role, a turning of its chemistry from a cause of death into a potential instrument of therapy, using the very specificity and potency that makes alpha-amanitin so dangerous to direct it precisely against cancer cells, while sparing the surrounding normal tissue, a concept that belongs to the broader field of antibody drug conjugates that has become one of the most active areas of oncological research in recent years. Clinical trials of amatoxin antibody conjugates in cancer patients began in the second decade of the 21st century, with early results suggesting that the approach had genuine promise in certain tumor types, particularly in cancers that expressed specific surface antigens to which the antibody component of the conjugate could bind with high specificity, and the early clinical data, while necessarily preliminary, was sufficiently encouraging to support continued development and further trials, the details of which have been published in oncological journals and have attracted the attention of both the scientific and the medical press. It is a genuinely striking thought, one worth sitting with for a moment in the quiet of this hour, that a molecule first encountered by human beings as the cause of accidental deaths in the forests of Europe, a molecule whose history runs through the courts of Roman emperors and the deaths of popes and the triggers of dynastic wars, should in the 21st century be dressed in the clothing of targeted medicine and sent into the bodies of cancer patients as a therapeutic agent, carrying its destructive chemistry to precisely the cells that need to be destroyed. A remarkable transformation in the moral and practical identity of a compound that has not itself changed at all. That is still exactly what it has always been. Still the same bicyclic octopeptide, still the same inhibitor of RNA polymerase II, still the same chemistry that worked its way through the bodies of the famous and the unknown dead across 2,000 years of recorded history. And perhaps there is something gently consoling in this, in the knowledge that even the most reliably lethal things that nature has produced may, in the hands of an imaginative and sufficiently skilled science, be turned toward the service of life, not as a denial of what they are, but as a redirection. A careful and technically demanding redirection of a force that was never aimed at anything in particular, and that can, therefore, with sufficient ingenuity, be aimed at something useful. The night is deep now, or it may be, wherever you are, wherever your body rests, and your breathing has settled into its slow and steady pattern. And the story of the death cap is deep too, deeper than we have gone yet, though we have come a long way from the pale and lovely cap rising from the forest floor to the clinical trials of amatoxin antibody conjugates in cancer centers in Germany and the United States, a long way through time and through the many registers of human experience that this single organism has touched. We have moved through ancient Rome and the deaths of emperors, through the medieval herbals and the courts of popes, through the wars set in motion by the death of a mushroom-loving Habsburg emperor, through the 19th century laboratories, where the first systematic chemistry of the fungus was attempted, through the post-war decades when Theodore Wieland and his colleagues finally cracked open the molecular structure of the amatoxins, through the clinical medicine of the 20th century and the development of psilocybin and the arrival of liver transplantation as a last resort, through the geography of an introduced species spreading along the root systems of European trees planted in California and Australia, and a dozen other places far from home, through the cultural history of the mushroom in art and literature, and the imaginative life of European civilization, and through the remarkable contemporary story of a deadly molecule being transformed into a potential medicine. There is more still, more history and more science, and more of the reflective and philosophical dimension that has threaded through all of this. And there is time still, and the voice will continue, and the story will continue, and the night will continue, and all of it will unfold at this same pace, this same unhurried, gently monotonous pace. That is not the pace of urgency, but the pace of deep time. The pace of a mushroom growing in the dark. The pace of a liver slowly remaking itself after injury. The pace of a scientific understanding accumulating grain by grain over the decades until it is dense enough and detailed enough to hold the weight of the problem it was built to address. in the forests outside whatever city or town or rural place you rest in tonight. There may or may not be death caps growing, depending on where you are and what trees are near you and what the season is. But somewhere in the world, in the oak forests of Germany and France and England and Italy, in the parks of Canberra and San Francisco and Washington, and 100 other cities, where European oaks were planted by people who did not know what they were carrying in the soil around their roots. The death cap is present in the ground, in its mycelium, patient and invisible, waiting for the season and the moisture and the particular set of conditions that will prompt it to send up its fruiting body, its pale and lovely cap on its slender stem, rising from the earth with the unhurried certainty of a thing that has been doing this for a very long time and will continue doing it long after any particular night, any particular voice, any particular listener has come and gone. And that is not a troubling thought, or it need not be, not in this hour, not in this quiet, because there is a kind of peace in the awareness of things that are very old and very patient and very much themselves, things that do not need anything from us, that proceed according to their own nature, that are not waiting for our attention or our understanding, but simply are, in the way that the forest simply is, and the rain simply is, and the slow turning of the season simply is, and all of it can be held gently, at a comfortable distance, from the warmth and the dark and the rest of a night that asks nothing of you now but to breathe and to let go and to sleep. There is a particular quality to the hours that come after midnight, a quality that those who have spent many nights awake will recognize without needing it described, a quality of the world having grown quieter than it was before, quieter than it was even at 11 or 12, as though the night has settled more deeply into itself, found a lower register, and those who are still awake in it find themselves in a strange and intimate companionship with the darkness and with whatever voice or thought or sound is keeping them company in it. It is in this register that we continue our story, moving now into what we might call the modern era of death cap science and medicine. The period from roughly the 1990s onward, when the tools of molecular biology and genomics and clinical research were being brought to bear on the problem with a precision that earlier centuries could not have imagined, and when the death cap was revealing new dimensions of itself to those who looked carefully enough. The 1990s were a decade of extraordinary transformation in the biological sciences, driven in large part by the development and rapid maturation of molecular biological techniques that allowed researchers to read and manipulate the genetic material of living organisms with a speed and accuracy that had been entirely beyond reach only a generation earlier. And these techniques opened new avenues for the study of the death cap and its toxins that supplemented and in some ways transformed the older chemical and clinical approaches. One of the most significant developments of this period, from the perspective of the death cap's history, was the beginning of serious genomic and transcriptomic work on Amanita phalloides itself, work that sought to understand not just the structure of the amatoxins, but the genetic machinery by which the mushroom produced them, the specific genes and enzymes that were responsible for assembling these extraordinary cyclic peptides from their amino acid building blocks in the cells of the fungal tissue. The question of how the death cap synthesizes alpha-amanitin is one that occupied researchers for many years because the biosynthesis of cyclic peptides is in general a complex business, and the specific pathway by which Amanita phalloides constructs its amatoxins turned out to involve a class of enzymes called NUBS, independent peptide synthetases of a type that had not previously been well characterized in fungi. And the full elucidation of this biosynthetic pathway required the development and application of genomic sequencing methods that were not available until the early 21st century. The work was eventually carried out by a team led by Jonathan Walton at the Michigan State University Plant Research Laboratory, who published in 2007 a landmark paper identifying the specific genes responsible for amatoxin biosynthesis in Amanita. Phalloides, a discovery that for the first time explained at the genetic level how this mushroom makes the compound that kills, and that opened new possibilities for understanding the evolutionary origins of the amatoxin biosynthetic pathway and the reasons why it evolved in the first place. The evolutionary question is itself a fascinating one, because from the perspective of natural selection, the production of amatoxins at metabolic cost to the fungus must confer some benefit that outweighs that cost, and the most plausible hypotheses involve defense against the many organisms, from insects to nematodes to grazing mammals, that might otherwise consume the mushroom and reduce its reproductive success. Though demonstrating this experimentally has proven difficult because of the challenges of working with a fungus that cannot be readily cultivated in the laboratory. The elucidation of the amatoxin biosynthetic genes also opened the possibility, still being explored in ongoing research, of engineering the pathway into more tractable organisms to produce amatoxins in larger quantities for research and therapeutic purposes, and of engineering variants of the pathway to produce modified amatoxins with potentially altered or improved properties for use in antibody drug, conjugates, and other therapeutic applications. And so the genomic era brought the death cap fully into the 21st century of biological science, not just as a clinical problem and a historical curiosity, but as a subject of active fundamental research, a research subject whose unusual biology and extraordinary chemistry continued to generate new questions and new insights. Decades after the basic structure of its toxins had been solved, the clinical management of Amanita phalloides poisoning in the early 21st century represents the accumulated wisdom of more than a century of careful medical observation, combined with the results of formal clinical trials where they exist, and the clinical experience of centers in Europe, North America, and Australia that have seen sufficient numbers of cases to develop genuine institutional expertise. The standard of care as it existed by the second decade of the 21st century involved several components that worked in complementary ways, each addressing a different aspect of the poisonings clinical course. And it is worth describing this standard of care in some detail because it reflects a genuine achievement of medical science, the transformation of a condition that was essentially untreatable in any specific sense, as recently as the mid-20th century, into one for which a rational and evidence-based approach could be offered, even if that approach remained imperfect, and the outcomes in severe cases remained uncertain. The first component is early diagnosis, which depends on obtaining a careful history from the patient or from anyone who accompanied them, a history that includes, specifically, whether wild mushrooms have been consumed, when they were consumed, and if possible, what they looked like. And this information, when obtained, can prompt the immediate initiation of treatment before the clinical course has progressed to the point of irreversible liver damage, which is why emergency physicians who see patients with gastrointestinal symptoms in areas where Amanita phalloides grows have been trained and retrained to ask specifically about mushroom consumption as part of their initial assessment. The second component is gastrointestinal decontamination, which in the form of activated charcoal can still be useful if administered within 1 to 2 hours of ingestion, binding any remaining toxin in the gastrointestinal tract before it can be absorbed. Though, as we have noted, the deceptive latency of amatoxin poisoning means that patients rarely present to medical attention within this narrow window, and the utility of activated charcoal in practice is therefore limited to the relatively small proportion of cases where someone recognizes very quickly that a potentially dangerous mushroom has been consumed and seeks medical attention before the onset of any symptoms. The third component is intravenous psilocybin, administered in high doses for several days, which, as we have discussed, may reduce hepatocyte uptake of the toxin if given early enough, and which has become a standard treatment in European centers and is increasingly available in North America and Australia as well, though access to intravenous psilocybin has historically been uneven. Because the compound was for many years available as a licensed medication only in Europe and had to be obtained through compassionate use or emergency protocols in other parts of the world. The fourth component is aggressive supportive care, including careful management of fluid balance, electrolytes, blood sugar, and coagulation, and close monitoring of liver and kidney function, with the aim of keeping the patient as stable as possible while the extent of organ damage becomes clear and while the body either recovers spontaneously, as it does in a significant proportion of cases where the dose of toxin ingested was not sufficient to cause irreversible damage, or progresses toward the need for more definitive intervention. The fifth and final component, reserved for the most severe cases where liver failure is clearly established, and the prospect of spontaneous recovery appears remote, is liver transplantation, a major surgical procedure that carries its own substantial risks, and that requires a donor organ to be available within a narrow time window, and that has been performed in several hundred cases of mushroom poisoning worldwide over the past 3 decades, with outcomes that have generally been better than those achievable with supportive care alone in the most severe cases. Though the decision to proceed with transplantation in a patient with mushroom poisoning involves complex clinical and ethical considerations that are not always straightforward to navigate. The decision criteria for transplantation in amatoxin poisoning have been the subject of ongoing refinement, with several scoring systems proposed and tested in different clinical series, trying to identify at the earliest possible moment which patients are likely to require a transplant and which may recover with supportive care, because transplanting a patient who would have recovered naturally would expose them to unnecessary risk, while delaying transplantation in a patient who needs it urgently may result in death from liver failure before an organ can be found. The development and validation of these scoring systems has required the accumulation of clinical data from large series of cases, data that has been collected primarily at specialized centers in Germany, France, Italy, and Switzerland, where the volume of mushroom poisoning cases has been sufficient to generate the statistical power needed to test and refine the criteria. And through all of this careful and painstaking clinical work, the underlying message has remained consistent and somewhat humbling, which is that the best treatment for death cap poisoning remains prevention, remains the accurate identification of mushrooms before they are consumed, remains the maintenance and transmission of the knowledge that distinguishes the dangerous from the safe in the woodland environment, a knowledge that is now more important than ever as the death cap extends its range to new parts of the world and encounters new populations for whom that knowledge is not part of their cultural inheritance. Prevention, in the context of Amanita phalloides poisoning, is a subject that occupies public health professionals and mycologists and community educators in many countries, and it is a subject that is at once simple in its essential message and genuinely complex in its practical implementation for reasons that we have already touched on and that are worth exploring in somewhat more detail here, because the complexity of prevention is not merely a logistical challenge, but a cultural and epistemological one, a challenge that goes to the heart of questions about how knowledge is transmitted between generations and between communities, and how the authority to identify and classify the natural world is established and maintained in different cultural contexts. The essential message of prevention is simple enough. Do not eat wild mushrooms unless you are certain of their identity. And if you are not certain, do not eat them. And if you have the slightest doubt, consult an expert before consuming anything you have foraged from the wild. This message has been communicated through many channels in many countries, through public health campaigns, through social media, through community outreach programs targeting the immigrant communities most at risk, through mycological society forays and identification workshops, through warning signs in parks and nature reserves, where death caps are known to grow, and through the education of emergency physicians and poison control staff. The complexity enters when then this simple message encounters the reality of how people actually relate to wild food and to the knowledge of the natural world, because for many communities, the harvesting of wild mushrooms is not merely a practical food gathering activity, but a deeply cultural one, embedded in family tradition and seasonal ritual, and a particular kind of embodied knowledge that passes from grandparent to grandchild in the forest, in an act of gathering and cooking and sharing, in the specific sensory vocabulary of a particular landscape and its particular species. The Hmong communities of California, the Chinese immigrant communities of the Bay Area, the Vietnamese families of New South Wales, the Southern European immigrants of Montreal and Toronto, all carry with them foraging traditions that are genuinely valuable within the ecological contexts in which they developed. Traditions that represent accumulated wisdom about the edible and inedible fungi of particular landscapes. And the challenge is not that these traditions are wrong or uninformed, but that they were formed in landscapes that did not contain Amanita phalloides, and that the translation of foraging knowledge from one landscape to another requires not merely the addition of a new warning, but the revision of a deep and embodied pattern of recognition, a revision that is psychologically and culturally far more difficult than it might appear from the outside. Public health campaigns that present simple warning images of the death cap alongside messages to avoid it are genuinely useful for those who encounter them with open attention, but they cannot easily override the confidence that comes from years of successful foraging in a familiar landscape, the confidence that says, I know what this looks like and I have eaten it before, and it was fine, a confidence that is entirely rational within the context that formed it, and entirely dangerous in a new context that contains a species not present in the original one. The work of mycologists and public health educators who have thought carefully about this challenge has moved in recent years toward approaches that are more culturally sensitive and more ecologically specific, approaches that acknowledge the genuine expertise of traditional foraging communities while providing the specific additional knowledge needed to avoid the one or two species that pose the most serious risk in their new environment. Approaches that involve working with community leaders and cultural organizations, rather than broadcasting generic warnings, and approaches that frame the issue not as a matter of ignorance to be corrected, but as a matter of ecological difference to be navigated, an important distinction that makes the educational message more likely to be received with the openness it requires. In Australia, the work following the 2023 deaths included engagement with Vietnamese and Chinese community organizations in Canberra and Sydney, with materials developed in consultation with community members and made available in Vietnamese, Cantonese, Mandarin, and other languages spoken in the communities most at risk. A more careful and culturally attentive approach than the generic public warnings that had preceded it. And early assessments suggested that this approach produced better awareness outcomes than the broader campaigns that had come before. Meanwhile, mycological societies in Australia, the United States, the United Kingdom, and many European countries have been working to make their expertise more accessible to the general public, offering identification workshops and foray programs that provide practical training in the visual and tactile features of edible and toxic species. And while these programs cannot reach everyone who might be at risk, they have contributed to a broader culture of informed foraging that includes a more widespread awareness of the death cap and its dangers than existed a generation ago. And through all of this prevention work, the mushroom itself remains in the ground, in its mycelium, unaffected by the warnings and the education campaigns and the public health infrastructure that has grown up around it, doing exactly what it has always done, waiting for the season and the rain. The question of what the death cap tells us about the relationship between human beings and the natural world is one that does not have a simple answer, and it is the kind of question that is best approached not with the urgency of a conclusion to be reached, but with the patience of a thought to be unfolded slowly, the way a piece of cloth is unfolded in the morning light to reveal a pattern that was not visible when it was bundled and compressed in the dark. The death cap is not a malevolent entity, as we have noted, but neither is it simply neutral in any meaningful human sense, because its presence in the world has consequences for human beings that are real and serious, and have been real and serious for as long as human beings have been walking through the forests where it grows. And the history of those consequences, the history we have been tracing through these many chapters, has a shape and a character that is worth attending to as a whole. One of the things that shape reveals is the remarkable persistence of the gap between knowing and preventing, between understanding a danger and reliably avoiding it. A gap that has not narrowed as quickly as one might expect, given the amount of knowledge that has accumulated around this particular danger over 2,000 years of recorded history. The Romans knew that certain mushrooms could kill, and they continued to die from mushroom poisoning. Medieval physicians knew that mushrooms were a source of danger and wrote about that danger in their medical texts, and people continued to be poisoned. 19th century mycologists named the death cap and described it with precision, and people continued to mistake it for edible species. 20th century toxicologists characterized its chemistry in molecular detail, and people continued to forage and consume it without adequate knowledge. 21st century public health authorities mounted extensive prevention campaigns, and the poisoning cases continued, including the California superbloom of January 2026. The persistence of this gap is not an indictment, of any particular individual or community or institution, but rather a reflection of a deep truth about the relationship between formal knowledge and embodied practice, between what is written in books or broadcast in campaigns, and what is known in the hands and the eyes, and the accumulated experience of a person standing in a forest making a decision about what to pick up. Knowledge of a general kind, even accurate and widely disseminated knowledge, is not automatically and reliably translated into the specific practical competence that safe foraging requires. And that translation requires not just the transmission of information, but the transmission of something more like skill, something that can only be fully acquired through practice and mentorship and repeated experience in the actual environment. And this requirement places a ceiling on what any campaign or warning system can achieve in the absence of that deeper kind of transmission. There is something in this that connects to broader questions about the limits of formal knowledge, about the things that resist being fully captured in propositional form, and that can only be known by doing, and about the particular vulnerability that comes from being in an unfamiliar environment with the habits and expectations of a familiar one, a vulnerability that is not unique to foraging or to mushrooms, but that the death cap illustrates with a particular clarity and a particular finality. At the same time, there is in the history of this mushroom a story about human resilience and adaptability, about the capacity to confront a persistent and genuinely difficult problem, and to respond to it with increasing sophistication over time, even if the response has not been fully adequate, and even if the problem has not been solved. Because the development of psilocybin as a treatment, the establishment of liver transplantation as a last resort, the creation of culturally sensitive prevention programs, the elucidation of the amatoxin biosynthetic pathway, the development of amatoxin-based cancer therapies, all of these represent genuine human achievements in the face of a challenge that is both very old and very stubborn. And perhaps the most honest and most complete thing that can be said about the relationship between human beings and the death cap is that it is an ongoing one, a relationship that is still being worked out, still being negotiated in forests and hospitals and laboratories and community centers and emergency departments, a relationship that has no final resolution, no moment of complete mastery or complete surrender, but only the continuing effort to understand and to adapt and to do a little better than before. There is a dimension of the death cap story that we have not yet fully addressed, a dimension that lies in the texture of individual experience, in the specific particular quality of what it is like to be a person in the midst of a death cap poisoning, not as a clinical abstraction, but as a human being moving through the hours and days of a condition that unfolds with a particular and terrible slowness. We have described the phases of the poisoning in medical terms: the latency, the gastrointestinal phase, the hepatorenal phase, and we have noted the clinical consequences of each. But the medical description, however accurate, does not fully convey the specific human experience of a condition in which the most dangerous thing is the absence of any signal, the experience of feeling entirely well for the hours after consuming something that is already invisibly in the process of destroying the liver's capacity to function. Physicians who have cared for patients with severe Amanita phalloides poisoning have described in published case reports and in interview accounts a particular quality of distress that comes not from the worst moment of the illness, but from earlier moment when the person, still feeling relatively well, is told for the first time what has happened, what they have consumed, what the likely course of events is, and what the options for treatment are. A conversation that must be conducted with both honesty and care, that must convey the seriousness of the situation without removing all hope, and that must also gather as quickly as possible the information needed to guide clinical decisions, including the identity of the mushroom, the quantity consumed, the time of consumption, and whether others who ate the same meal are also at risk, and if so, whether they have been located and are receiving care. The families of those who have been poisoned, particularly in cases involving children or in cases where multiple family members are affected, face a particular kind of anguish that is compounded by the knowledge that the harm was accidental, that there was no malice involved, only a mistake, often a mistake made by someone with genuine experience and genuine competence in a different ecological context, and the weight of that knowledge has been described by survivors and family members in terms that reflect both grief and a kind of complex self-understanding that resists easy resolution. The physicians and nurses who care for these patients carry their own particular burden, the burden of managing a condition for which the therapeutic options are limited and for which the outcome depends heavily on factors outside clinical control, including how much toxin was consumed and how quickly the diagnosis was made, and whether a donor liver becomes available in time, and the emotional weight of this uncertainty, of doing everything correctly and still losing patients, has been described in the medical literature and in personal accounts as a distinctive feature of the clinical experience of amatoxin poisoning management. Survivors of Amanita phalloides poisoning, those who consumed a sublethal dose and recovered with or without significant medical intervention, have described, in the accounts that have been published and broadcast, a range of long-term responses to their experience, from a deepened awareness of the fragility of life to a lasting wariness about wild food to a sustained engagement with mycological education as a way of transforming a traumatic experience into something that might benefit others. And the stories of individual survivors contain within them a human dimension that the clinical literature, for all its precision and its genuine value, cannot fully capture. The deaths that have occurred from death cap poisoning across the centuries and across the world represent an enormous accumulation of individual human stories, most of them unrecorded, most of them known only to the families and communities directly affected. And the named deaths that we have followed through this documentary, the emperors and the popes and the clusters of cases in California and Australia, are only the visible peaks of an iceberg whose submerged mass consists of the countless ordinary people who went into the forest on an autumn morning and did not come home in the same health they left in. People whose names are not in any history book, whose stories are not preserved in any archive, but whose experiences were as real and as significant as any event in the documented record, and whose collective weight adds a dimension of human meaning to the natural history of this mushroom that no amount of chemistry or taxonomy can fully substitute for. There is a practice among some mushroom poisoning researchers and public health professionals of maintaining records, not just of the clinical data of cases, but of the human stories behind them, the circumstances of the foraging, the meal that was prepared, the family that gathered around it, the hours that passed before the first symptom appeared, the phone call to the hospital, the ambulance, the days in the intensive care unit, and this practice of attending to the human story, alongside the clinical data, reflects a recognition that the full significance of what the death cap does in the world cannot be captured by numbers alone. that the history of this mushroom is at bottom a history of human experience, of the specific texture of encounter between one form of life and another, repeated across centuries and continents, with endless variation in the details and remarkable consistency in the essential shape. The question of what comes next, of where the story of the death cap and its relationship with human beings is headed in the coming decades and centuries, is one that can be approached only with appropriate humility, because the history we have traced through these chapters contains repeated reminders of how poorly human beings have generally predicted the future behavior of the natural world and the future trajectory of their own efforts to manage it. What the scientific evidence suggests, as of the present moment, is a set of trends that point in several directions simultaneously, some of them encouraging, and some of them concerning, and it is worth sitting with both sets without rushing to resolve them into a single simple narrative. On the encouraging side, the ongoing development of amatoxin antibody conjugates as cancer therapies represents a genuine possibility that this molecule, so long associated only with harm, may within the next few decades have an established role in the treatment of specific cancer types, a development that would represent one of the more remarkable reversals in the history of natural toxins, and that would add a final redemptive chapter to a very long and often devastating story. The improving cultural competence of prevention programs, the growing availability of intravenous cybinin in countries where it was previously difficult to obtain, the development of better clinical decision tools for the management of severe poisoning cases, the continued growth of the mycological community, and its public engagement activities. All of these represent genuine progress toward reducing the harm that the death cap causes. Progress that is real, even if it is slow, and even if its ultimate limits are not yet clear. On the concerning side, the continued range expansion of Amanita phalloides into new regions, driven by the ongoing planting of European trees in parks and gardens around the world, and by the possibility that climate change may be creating conditions favorable to fruiting in areas where the mushroom's mycelium has been established for years without producing visible fruiting bodies, suggests that the geographic footprint of the risk is likely to grow rather than shrink in the coming decades, bringing the death cap into contact with new communities and new ecologies in ways that will require continued adaptive response from public health and clinical systems. The increasing mobility of human populations, the continuing movement of people across ecological boundaries, the persistence of traditional foraging practices as both cultural expression and practical food gathering in immigrant communities around the world. All of these suggest that the particular pattern of risk that has driven the most recent decades of death cap poisoning epidemiology will continue and may intensify, requiring approaches to prevention and clinical care that are more culturally specific and more ecologically informed than anything that has existed before. And the fundamental challenge, the challenge of translating general knowledge into specific practical competence in the act of foraging, the challenge that has persisted throughout the 2,000 years of this story, shows no sign of becoming easier, because it is a challenge rooted not in the inadequacy of our knowledge, but in the nature of the knowledge that foraging requires, knowledge that cannot be fully transmitted through warnings and campaigns, but only through the slow and patient accumulation of embodied experience, and that therefore requires an investment of time and attention and mentorship that is not easily scaled to the pace at which populations move and ecologies change. And so we come, at last and very slowly, to the end of our story, though of course it is not the end of the Death Cap story, which will continue long after this voice has fallen silent, and this night has passed into the ordinary light of morning. We have travelled a long way together, through the ancient forests, where our earliest ancestors learned by trial and error which mushrooms could be eaten, through the courts of Roman emperors, where that learning was not quite sufficient, and the consequences were profound and lasting, through the medieval herbals and the Renaissance kitchens and the botanical gardens of the 19th century, through the laboratories where patient chemists spent years cracking open the molecular secrets of a cyclic peptide that nature had spent millions of years refining, through the hospitals and intensive care units where physicians worked and continue to work with the imperfect but growing toolkit of modern medicine, through the parks and suburban neighborhoods of California and Australia, where a European mushroom grows at the foot of an oak tree, planted by someone who did not know what they were also planting, through the community outreach offices and the Mycological Society forays, and the emergency departments of cities on 3 continents, through the cancer research centers, where the same molecule that has been killing people for 2,000 years is being carefully loaded onto antibodies and sent into tumors, and through the long and quiet philosophical territory that opens up when you follow any single thread of natural history, long enough and far enough to see how it is woven into everything else, into the history of empires, and the ecology of introduced species, and the sociology of immigrant communities, and the molecular biology of gene expression, and the deep and patient rhythms of the fungal kingdom that underlies and supports and outlasts almost everything that we, in our brief and energetic way, have managed to build and name and understand. The death cap will fruit again this autumn, as it has fruited every autumn, in the oak forests of Europe, and in the parks of Canberra, and the backyards of Berkeley, and the suburban streets of 100 cities, where someone long ago planted a European tree and did not think about what was living in the soil around its roots. It will rise from the ground in its pale and beautiful way, wearing its faint olive cap and its white vulva like a little skirt at the base of its stem, offering no warning, requiring no effort, simply present in the way that all natural things are simply present, in the way that the rain is present and the dark is present, and the slow turning of the seasons is present, without intention and without mercy and without malice, simply doing what it does because it has always done it, and because the chemistry and biology that it is made of will continue to do it for as long as the conditions that support it exist. And you are here, breathing slowly in the dark, safe from the forest and from the autumn, and from the cold soil where the mycelium waits, safe in the warmth that holds you, safe in the night that has received all of this history, and all of this science, and all of this quiet philosophical attention, and has carried it gently past you, not asking you to hold it or solve it or resolve it into anything, only asking you to let it pass the way water passes, smoothly and without resistance, leaving behind only a certain quality of having been in contact with something very old and very patient and very much larger than the span of any single human life. The story of the death cap is a story that began before history and will continue after it, and we have moved through only a small portion of it tonight. A small and carefully chosen portion, moving slowly enough to notice the details and quietly enough to hear the silence between them. And now the silence is growing larger than the words, and the night is moving toward its far shore. And the body that has been carrying you through all of this is ready, if you will allow it, to release the last of its wakefulness and settle into the deep and wordless rest that is waiting just below the surface of this moment, as it has been waiting all along, patient as mycelium, certain as autumn, soft as the forest floor, in the last days of the year, when the mushrooms have finished and the leaves have fallen, and the ground is quiet and dark, and full of the slow, invisible work of things that do not need to be watched to continue. Things that persist in the deep and steady way that life persists, through every season, and every century, and every long and restful night.